Welcome back, students. Welcome back to the fifth semester Medical Lab Technology Lab Practical. Today we'll discuss about estimation of erythrocyte sedimentation rate. This test which can be help out for you the patient anemic condition or polycythemia and any type of infection we can identify it because of the false level of the RBC. Mostly we can find out this test in the doctor's prescription slip. When the doctor make the prescription slip for the patient, they will mention the ESR. And any people are see the ESR, almost it's a finalized like this is the erythrocyte sedimentation rate. Mostly we can find out ESR only. Okay, when you see the ESR, you should identify it immediately ESR test, that is you need to perform. And today we can see what are the objectives we have, what we need to do in the next other slides. First of all, the aim of the test. Every test should be aim you need to write. Principle of the test. Stages of erythrocyte sedimentation rate. Methods of estimation of erythrocyte sedimentation rate. Westerveen method. We have the several methods. From the several methods, we need to learn universal accepted and relevant method, that is the Westerveen method. Today we will discuss about the Westerveen method. Under this method, equipment and reagent. What are the equipments required for this test performance? And specimen. What type of specimen we need to collect, and what is the process, how we can store the specimen. And next one, procedure, the test procedure, how we can perform the test procedure step by step. And result, precautions, reference ranges, and factors affecting on erythrocyte sedimentation rate. Aim to perform erythrocyte sedimentation rate for given or your own blood sample. If lab they will provide to you sample, you can do with the sample. If lab cannot provide, you need to collect your own blood sample. I think you people are expert in the blood sample collection because of the now you people are in third year. This is the ESR test. Okay, you can see this type of the RBC falls you can need to observe in this picture. Okay, what are we have the number of pictures also is there for observation of the ESR test and procedure purpose also. Let's see. Here we need to discuss about principle. The erythrocyte sedimentation rate measures the rate of settling or sedimentation of erythrocyte in anticoagulated whole blood. यहाँ पे हम blood में anticoagulant mix किया. Okay, when you people are mixed the anticoagulant is the blood, there is no clotting formation because of the anticoagulant present होते हैं. Then what happen, if it is the same blood, mixed blood, you can keep it in vertical stand. Okay, like whatever the blood composition RBC होते हैं, WBC होते हैं, and plasma होते हैं, and several minerals होते हैं, वो सारे automatically based upon the molecular weight and molecular side, it can be settled down very slowly. The anticoagulated blood is allowed to stand in a glass tube for 1 hour, and the length of column of plasma above the red cell is measured in millimeters. This is correspond to the ESR. And here stages of erythrocyte sedimentation rate. There are the 3 stages will be there. There are the 3 stages main important. The first one is the formation of relax or lag form. It will be take 10 minutes in vertical stand. The red cells stuck together like a pack of the coins. The ESR depends mainly on this stage only because the RBC is the dumbbell shape, and when it will be mixed in the like a tissue fluid, like a liquid like a plasma, it's freely flotation होते. हम whenever keep it in vertical stand without any disturbing, it slowly falls down. That is the रोलैक्स फॉर्मेशन बोलते हैं. You can see like next stage sinking. Okay, it's like after that rolex formation, then sinking together, it's bounding, it's started to bounding together, and it will be take 40 minutes. The first stage is 10 minutes, second stage 40 minutes. Rapid and constant sedimentation, the longer the tube, the longer is the stage, is the higher the value of ESR. Here we can observe the high level of the ESR falls. And next one, stages of erythrocyte sedimentation rate पे the third stage packing or Rolex slow sedimentation, 10 minutes, 40 minutes, again 10 minutes. Finally the RBC form like this one, the RBC form one by one, like packing like this one, like a coins, one rupee kinds of, when you keep it one by one isa, the same way the cells will be packing all RBC. Then because of the plasma have the low molecular weight, this is the only liquid, it's like a come to the upper, which are the having high molecular weight like RBC, it's settle down, the sediment bolte hum. Next, the methods of ESR, the methods of estimation of the ESR. You can see the Westergin method, second one Introbe method, third one Zeta sedimentation ratio, fourth one micro ESR. Mostly the accepted India पे हम Westergin करते, Wintrobe कभी कभी, micro ESR हम depending on the hospitals or laboratory. Today we will discuss about this method, that is the Westergin method for estimation of ESR. The International Council for Standardization in Hematology recommends Westergin method for estimation of ESR because this is the accuracy result you will get it, like 100% is very good. This is the Westergin method, tube and stand. Okay, this is a measuring tube and also one sample tube bolte. This is the stand. We need to like a marking like this one only in the laboratory. Equipment and reagents. The first one, the Westergin ESR tube. How it will be appear the ESR tube? This is the straight glass pipette measuring 300 mm in length and calibrated in mm from 0 to 200, top to bottom, 0 to 200. You can see this picture, 0 to 200. Then the marking are only over the lower 2/3 of the tube. The tube is open ended, both sides it can be ended, open part, and internal diameter should be less, this should not be less than 2.5 mm. The tube should be clean and air dry before using. This is the picture, it will be showing the Westergin tube. You can see 0 to 200. Okay, total tube length 300 mm. Westergin stand, this holds the tube in the motionless vertical position. Motionless means without disturbing, we need to stick the stand. Okay, the stand appear like this one. The stand is depends upon the company. Individual companies, individual stands will vary. This is one of the example for the Westergin. Without disturbing, is the standardized fixed. Something if you are during the test, if you are disturbing the stand, automatically you cannot get the proper report, then it's like a false report. Please mind it, you need to do not disturb this stand and after loading tube with the blood. Equipments, anticoagulant dilution solution, that is the 3.8% sodium citrate. How we can prepare the sodium citrate 3.8%? Measure trisodium citrate dihydrate 32.08 grams mixed in the distilled water. It should be 1000 ml. Then this distilled water you can store at the 4 degrees Celsius. Okay, before using you need to filter the with the membrane, sterile membrane 0.22 microns you need to use it. And after sterile you can go for the sterile means this is the filtration, then you can go for the storage during at the 4 degrees Celsius. After storage you can utilize this solution several months. Okay, but if it becomes turbidity, if you find any turbidity nature inside of the liquid, then you need to discard, because of there is the fungus will be growth or molds will be growth. That's why you need to go for the discard. And alternatively, instead of this trisodium citrate, we can use EDTA sample also, EDTA tube also. This is the EDTA tube. Specimen, the venous is collected in trisodium citrate in 4:1, 4% blood, 1% citrate, sodium citrate portion. If the specimen is kept at room temperature, test should be carried out within 4 hours of blood collection. If stored at 4 degrees Celsius, a delay up to the 6 hours permissible. The blood anticoagulate with the EDTA can be tested within 24 hours if stored at the 4 degrees Celsius, provided it is diluted with the trisodium citrate before the testing. These are the sample. The blood sample you can use the aseptic condition only. I think you know very well how we can collect the cubital vein blood sample. Like that one we need to collect. The specimen, the steps here, the diagrammatic steps will be available here. You can see the first one, by the aseptic techniques we need to collect the blood sample, around 2 ml of the blood sample. Then after collection immediately you can go for the 2 techniques is there based upon the patient prescription. One is directly you can keep it the venous blood sample in the you can see first they are using the sodium citrate, few drops, then we can add the blood. This is the one step. And the same thing, the same blood you can add from the syringe directly we can add in the EDTA tube. These are the both steps. Okay, this EDTA tube also we can go for using for the ESR test, and this trisodium anticoagulant also you can go for the EDTA test. You can see after trisodium citrate, this EDTA tube, blood you can open the cap, then you can add in the EDTA pipette. Okay, this one. Then the final step, directly you can take the ESR tube and fix it like this one. You can fix it like this one. Here the same procedure I am going to repeat the point by point. The mixed anticoagulated blood sample thoroughly. The Westergin tube is filled with the blood sample up to the 0 mark. 0 mark means from the top. 0 mark is from the top. Bottom pe 200 mg. Up to that you need to fill it with anticoagulant bill without any air bubble. Without any air bubble. The tube is placed in the strictly vertical position in the ESR stand and let the undisturbed 1 hour, and it should not kept in direct sunlight and should not be subjected in the vibrations, like near to anything vibration like a centrifuge machine, and nearby windows we cannot keep the ESR stand. When you are loaded the ESR tube, immediately you need to note the time. After exactly 1 hour, read the height of the column of the plasma above the red cell column in mm. This one. You can see after 1 hour the RBC will falls like this one. the plasma from the 0 to up to the plasma कितना मार्क तक है वो इतना हम रीड करना पड़ेगा. देन फॉर एग्जांपल 5 mm 10 mm per hour 20 mm per hour. If it is patient have the tuberculosis severe infection, maybe 50 mm per hour, maybe patient have the severe anemic condition, maybe 20 mm per hour, sorry 180 mm 150 mm 20 mm aisa it will depending on the patient clinical condition you will get the result. The result depending on the your test results you need to give whether it is 5 mm or 20 mm per hour. Precautions: use the correct proportion of the blood and anticoagulant like a 4:1. Mix the blood sample anticoagulant thoroughly. There should be no clots and air bubbles in the blood, please mind it. The reference ranges relates to test performed at the room temperature of 18 to 25 degrees only. Don't go for very hot because of the if it is temperature is very high ESR will be increased and different reference ranges will be have the delivered. Matlab, when you are keep it the hot temperature automatically result will be different will get it. ESR tube must be kept vertically, even a slight tilting will be increase the ESR. Tilting matlab कोई भी angle, if it is 90 degrees, if you keep it like 70 degrees or 80 degrees or like 50 degrees, you will get the increase of the RBC levels. These are the reference ranges of the ESR. The ESR sedimentation rate by Westergaard method, reference method. The reference values will be changed by the based upon the methods, micro method, Winthrop method से अलग-अलग होते हैं. ठीक है. The males 50 years 0.15, female 0 to 20 because of the females have the higher values because of they have the less RBC count. The children 0 to 10 and elderly male 0 to 20 mm in hour, female 0 to 30 mm per hour. Due to the menstrual cycle females have the and also most of the females have the anemic infection, anemic condition, not infection, anemic condition. The factors affecting on the erythrocyte sedimentation risk. The ESR increase and decrease two factors available. Increase in the factors old age, pregnancy, anemia, availability of the fibrinogen, macrocytosis, technical factors high temperature, tilting of ESR tube. And factors decreasing on ESR, microcytosis, low fibrinogen, polycythemia, marked leukocytosis, technical factors vibration tube during the test. These are the practice questions for you. You can prepare these questions. Maybe you will get in examination these type of questions. Apply your knowledge on ESR, discuss on stages of ESR, illustrate principle and procedure of ESR, write full form of ESR, apply your knowledge on clinical significance, and what are the methods we can use for the estimation of ESR. These are all the questions. Once you can read this PPT, it can be help to you preparation of your examination. These are the reference textbook. You can get this same material from these three textbooks like Anatomy and Physiology of the Ashalata, and Medical Laboratory Technology by Gatkar, and Medical Pathology by the Harsha Mohan. Thank you, students. If you have any queries, please contact me. There is my mail ID and also my mobile number also is there. Anytime you can call and you can clarify your doubt. Thank you.